Hello, everyone, and welcome to today's webinar, USP Standards to Support AAV Quality Testing and Analytical Characterization of AAV Content Utilizing Mass Photometry and Orthogonal Techniques. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Refine. To learn more, visit refine.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speakers for today's webinar. Dr. Anthony Blaszczyk, Senior Scientist 2, United States Pharmacopeia, Dr. Lauren Tomlinson, Scientist, Pharmaron Biologics, and Dr. Paul Getty, Senior Technical Specialist, Pharmaron Biologics. Dr. Blaszczyk, you may now begin your portion of the presentation.
Hi, thank you for that introduction, and thank you for this opportunity to give this talk to everyone here today. As I'm sure you guys have all already know, I'm here today to talk about USP standards, specifically those related to AAV quality testing and characterization. So a lot of people are familiar with USP, but in these talks like that, a lot of people aren't at all. So we actually get a pretty big range of people. So what I'm going to try to do is give a quick enough introduction for those who are very familiar with USP. I don't spend too much time and bore you, but for those who are not too familiar, I can try to my best to catch you up in one slide. So USP, or US Pharmacopeia, is a standard setting organization. We are nonprofit, non-government affiliated. So unlike a lot of other pharmacopoeias in the world who are directly related to that country's government, we are completely independent. With that being said, we do work closely with the FDA because a lot of people rely on the USP for regulatory filings. We want to make sure we align with what the FDA is looking for, but we are both independent organizations, with USP being completely government independent. So as I said, we're a standard setting organization, and there's a lot of benefits to standards, including the three shown right here, being consistency, meaning that especially physical standards or documentary or method standards that help ensure that different methods are running properly or working properly, whether that's via system suitability that we offer or physical standard. Also, innovation. Having a well-characterized physical standard allows for new innovative technologies. And the way, one example of how this works is if a company were to develop a new technology and they were to test it with some material that they buy, a lot of people are going to be hesitant to really truly believe that the data they're getting is true, is accurate and reliable. However, if they can show that they're getting the same data, same values as on the certificate, of a USP standard, just using an orthogonal method, it's really going to add a lot of credence to their technology. And last is support. Our standards support a lot of regulatory filings, makes getting into the makes filing a lot easier, makes validations easier, and just makes a lot of analytics easier. The USP has three different types of standards. For those who are not too familiar with USP, I can almost ensure you that you are somewhat familiar with our monographs. So our monographs are typically for small molecules, and those are for a lot of different drug products out there to ensure that they meet the USP quality. So if anyone takes a look through their medicine cabinet, I'm sure they'll be able to find multiple different prescriptions or over-the-counter drugs that have the comma USP after that. That just means it's USP standard, and that particular drug is meeting the USP standard, which is usually for those type of drugs found on a monograph. We also have documentary standards or general chapters. There are two types of these. One are going to be greater than 1,000 chapters, which are going to be just kind of guidance, best practices. And then we have less than 1,000 chapters, which are going to be more like SOPs or step-by-step instructions. These less than 1,000 chapters are validated methods. They're recognized by regulatory bodies, and makes it much easier because people who use those methods don't have to go through a full validation. They can just do a verification. Last is physical standards, and these, as the title describes, are physical standards. They're usually well characterized in a multi-lab study. The heart of the talk today is going to be about a number of physical standards we developed, but also talk about a few of the general chapters. So in cell and gene therapy, we have a number of chapters that are relevant. However, a lot of people don't really realize that and don't think of USP when they think of cell and gene therapy. But we are trying to grow here. We've grown a lot over the past few years, as I'll touch on in the next slide. We're really trying to expand our footprint in this area. Shown on the left are some more not CGT specific, but just common analytical methods that people, if they don't follow directly, follow indirectly when doing a lot of analytical work. So things such as guidelines for validation, guidelines for verification, dealing with analytical methods, whether that's a complete life cycle analysis or transferring from like AD to QC. And then the right, we have a few specific CGT related general chapters. 1046 is cell and tissue based products, and 1047 is gene therapy based products. And lastly, another very interesting chapter is 1043. This relates to ancillary materials or starting materials, which anyone in the cell and gene therapy field knows are absolutely critical. Last part of this slide I'm going to direct your attention to is the bottom here, and what we call these are compendial methods. These are used for all different type of drug products, whether it's large biologics or small antibiotics. And these are routine tests everyone's running. I shouldn't say routine, I should say more common tests everyone's running. And oftentimes people don't even know they're using a USP method. These examples include pH, color, clarity, osmo, sterility, mycoplasma. These are things that all the labs are running just to ensure the safety of their products. They're very well established, and a lot of times people are just using a USP method for this. Moving on to USP's trajectory in the cell and gene therapy field. So we started doing work way back, even before 2020. We formed a Bio5 expert committee, and this is kind of a governing body at USP who approves chapters. And this Bio5 actually approved two different chapters, as I mentioned, that were talked about on the last slide, relating to cell and gene therapy. This was all pre 2020, and then from 21 to 2023, we really started ramping up our work in cell and gene therapy. As you can see here, these are some just of the highlights. In 2021, we held an AAV roundtable. What we did here was gather a lot of stakeholders together in the same room to really talk about the needs and wants of this AAV community. This means what chapters are needed, what type of guidance is needed, what type of physical standards are needed for this field. Nimble, NIST, and USP also joined collaborated for an AAV collaborative study where we investigated empty full using a number of different methods. This was a multi-year investigation with a number of different labs, and this study was actually just published earlier this year. 2022, we formed a specific expert panel to create a draft chapter for best practices for AAV. Also, USP worked with the APEC, or the Asian Pacific Economic Coalition, to do COE training for CGT bioassays. 2023, USP and ATC joined forces to generate genomic DNA reference standards. I'll talk a little bit more on coming slides. Also, USP hosted, in collaboration with ARM, or Analytical Reference Group, a workshop on AAV CQAs. And then the standard coordinating body in USP and the FDA held a workshop on methods for harmonization, also in 2023. And then the last 2 years, as I mentioned, that Nimble NIST USP empty full AAV paper was published. We also released a plasmid DNA chapter for public comments online that was drafted by an expert panel. And then we launched an AAV online analytical guide, which we kind of talk about being everything you need to know about AAV analytics all on one website. It's an interactive website, for very user-friendly for people to go through and kind of see what's needed for AAV. And I'll talk a little bit more about this in a later slide as well. And in 2025, this past calendar year, we've really launched our AAV package for reference materials with a big focus on physical reference standards. And that's going to be the main focus on the rest of the slides. Go through this total package and talk a little bit about each of these standards that we're now offering. First thing we had to do when deciding what AAV standards is kind of figure out what are needed. And I talked about a few years ago, we held an AAV roundtable. We got ideas from stakeholders. We go to a lot of conferences. We talk to a lot of different companies, organizations, academics, instrument manufacturers, CMOs, CROs, CTOs. We talk to everyone, any leader in the field we try to talk to and get an idea of what they think is needed in terms of standards. And then usually by talking to enough people, we see the overlap, and it's not like an official survey, but we take unofficial surveys and figure out what's most needed in the field. So when looking at AAV, we had a focus on three main things to start on a high level. Upstream. These could be think of as raw materials such as plasmids. CGT and AAV in particular require novel raw materials, and the control of these raw materials is very essential for both the safety and um, production of these AAV molecules. We also focused on downstream or critical quality attributes. So this could be directly related to the impurities as well, which is shown on the right. So it's key for downstream to make sure you purify the right AAV, make sure that viral vector is in the proper form, make sure it's the correct material you want. So there's a lack of harmonized test methods for these. That's one thing we want to focus. There's also a lack of quality viral vector standards on the market, such as AAV reference standards. And then with impurities, as I talked about, you want to make sure you have that pure viral vector. There are also a lot of common impurities. So what we did, we're also focusing on our methods and physical standards to help test for those purities, because a lot of these starting materials for AAV production can be safe, can cause safety concerns. So it's very essential to remove all these starting materials, and residual impurity testing can be very challenging. So developing both physical and documentary standards for some of these impurities can lead to can really help the AAV field in a number of different ways. So shown on this slide is our AAV. First, we're calling this phase one of our AAV solution package. This includes a lot of key solutions to help address some of the most difficult analytical challenge people in this field are facing. So we have 7 original, we'll call solutions. Some of these will have more than one product. First one, just going from left to right, top to bottom, we have plasmids for qualification. These will be with a focus on the raw material, making sure your plasmids are of proper topology and a physical material for test them. Residual plasmid, that's for testing impurity of residual plasmid. Endonuclease, that will be another raw material to ensure the activity of your endonuclease. And residual host cell DNA, another residual impurity test. And then we have a few of the actual viral vector itself and different standards to help for different tests that assess the quality of your viral vector. And these 3 at the bottom, that's capsid quantification, AAV empty full ratio, and genome titration. And as I said, the rest of the talk will be focusing more on these, and each one of these will have its own time that I'll be able to talk about and hopefully get into more detail. And if I don't get into enough detail because of these, just for time restraints, feel free to ask me about any of these after this presentation. So first one I'll talk about is our AAV empty full reference standards. These are what we're calling our flagship product to start. These were actually just released in the last week of April, so they're very, very new to the market. And these are 2 different standards. They're both AAV8 serotype. One is just empty capsid standards, and one's full capsid standards. The capsid titer for the empty and full is 3.1 and 4.7 e to the 12 respectively. And both of these have 300 microliters, and our empty is aimed at being less than 10% full, while our full capsids are aimed at being greater than 90% full. It's critical to ensure the quality of these, especially if they're going to be used for standards by analytical labs. So we think there's a lot of advantages of our USP reference standard. This is the one standard that, talking to everyone in the field felt there was a need for new and improved AAV reference material. There are some commercially available reference materials or pre-made AAV out there. They just weren't of the quality, at least a lot of our stakeholders felt, to be truly considered reference material or reference standards. So I have a little table on top in the blue of why we think our USP product, how it separates itself from a lot of the other commercially available AAV material on the market. So first is ours is high volume. It's 300 microliters as compared to usually about 100 to 150 microliters per vial, and a high concentration. Ours at most, or they're both greater than 3 e to the 12 capsid particles, and the other one is 3.1, one is 4.7. So that's a much higher concentration than you typically see for pre-made AAV. And the reason these first two are critical is because some of these AAV physicochemical methods, specifically for empty and full characterization, require a high amount of AAV particles. So you need both a high volume and a high concentration in order to test some of these, such as like AUC or SEC mols. So if you're using other material out there, you're going to have to buy multiple vials, pool it, which is just extra steps that can introduce air to your testing. Some other advantages we have is we use multiple methods. So ours is analyzed by 5 separate empty full methods, where usually if you buy another product out there, it just only has one method. And anyone in this field knows that all these empty and full methods vary greatly. Just the science is different between all of them, and the technology used can lead to different values. And what people are seeing is they're buying like an AAV material and it has a empty full percent of, let's say, 80%, and they're getting 70%. They don't know if the material isn't what they said. They don't know if it's different in testing methods, or if they're just getting the wrong value. So by being able to provide test these by 5 separate methods, and be able to provide 5 separate values, we think we're doing a great service to the field by providing them an array of testing values that are most common in the AAV field. We also have this is also a multi-lab study. So 5 labs were involved with this. Most of these methods were tested by multiple labs, whereas other material often is just tested by a single lab, and you don't really get an idea of any variability. If a test was bad, you wouldn't really get that flagged at all. So these are all the advantages we feel like the USP standard has. This is just in regard to the empty full testing. We also did a lot of additional testing on this, including peptide mapping, SEC analysis, purity analysis, capsid titer, genome identity. Most of these are included on the certificate that you can find online. Some of these are we're still finalizing it, doing some review, and we'll add those to the certificate in the near future. So as I said, these standards are now available, so we can go through some of the data for these. As I said before, 5 different methods. Here's a value for these. On the table on the right, we have the percent full for the full, which would be the middle column, and then for the AAV empty, we have the percent empty. And as you can see, by all the methods, the percent full are all generally high, probably averaging about 90%. CDMS is the lowest at 80%. And most of this, we'll talk about a little bit on the next slide, is kind of deciding how to quantify the intermediates. Some methods have the ability to not only measure empty and full, but also measure intermediate species. Kind of drawn that line of where you call a partially filled or intermediate versus full can be a little challenging and not going to be an exact science. We also have the AAV8 empty on the right. That's a little more clear, and you can see all of those are over 91% empty. Give you guys a minute to just kind of look at these data and see how all the numbers compare. One thing's definitely clear is that we believe, and that's these data suggest that we do have high quality AAV full and empty. As I mentioned, there's also that partially filled AAV, and that is becoming more and more critical as we learn more about these AAVs. New studies are showing that these AAVs are actually quite heterogeneous, even the most what we'll call homogeneous of the AAV. And learning more about these partially filled AAV is also critical. So some methods that are very novel have the ability to test this partially filled AAV, and these methods really separate themselves from some of the other ones because of the criticality of this partial filled species of AAV. So in our study, the 3 methods that we're able to do that is mass photometry, labeled MP here, CDMS, as well as AUC. For those not familiar, CDMS is charge detection mass spectrometry. It's a specialized high molecular weight mass spectrometry. And AUC would be analytical ultracentrifugation. And again, just extra value here with being able to look at the percent of partially filled AAV for the empty and full AAV, both these standards. And you can see the CDMS for the full is definitely the highest, but we think looking at these data a little bit closer, and for all of these in this one, we just think it's not necessarily that the testing is so much different that we're getting completely different values. It's just kind of where drawn the line for what you're calling full versus partial. So this is our AAV solution, or empty full product here. As you can see, a lot of testing was done, and I was only able to get into the empty full testing and not touch on all of it. And as you can see, it's a lot of data that we really think will be able to help a lot of pretty much anyone who's studying empty full AAV particles. We have two more AAV standards I'd like to touch on, our genome titer and capsid titer or capsid quantification reference materials. The first one is our USP capsid titer reference materials. What this is really meant for in a 1-second elevator pitch is to normalize ELISA data. That, again, talking to stakeholders, people are having a lot of problems with their ELISA data. However, when everyone keeps going back to do ELISA, it's by far the most common capsid titer method out there, but people are seeing a lot of variability from kit to kit, whether that be lot to lot of the same manufacturer or using different manufacturer, getting different values. So USP has developed five standards, which are used to hopefully normalize ELISA data. What's unique about these is these standards were the capsid titer value were directly quantified using SEC mols. And this is a direct measurement, whereas ELISA is going to be kind of an indirect measurement because the value of an ELISA is going to be completely dependent on a standard curve or a kit calibrant. So if by any chance this kit calibrant was not properly or not accurately quantified, every value, even if you do the ELISA test perfectly and everything else is perfect, your values are going to be inaccurate because it's only as accurate as the kit calibrant. So by developing standards that were directly quantified using a direct measurement such as SEC mols, which isn't typically used for ELISA kit calibrants, we think this will really help align different ELISA kit values, or whether that's from kit to kit or manufacturer to manufacturer. So the serotypes we're starting that will be released are 1, 2, AAV 1, 2, 5, 8, and 9. And as you can kind of see here, what we envision happening, I'm not saying it's always going to be higher or lower, but if you have two different calibrants, and even if you have the same absorbance for your sample, that the value you're going to get is going to be completely dependent on the calibrant you use. We do have some preliminary data for this that I can share now. This was just done at USP, and this isn't something that's going to be shared on the certificate at this time. But what we have found is that if you use multiple different we did 4 kits in this experiment, but our USP standards can help normalize values from different ELISA kits. So in this experiment shown here, we used 4 different kits, and on one experiment we measured them with the kit calibrant. On the second experiment, we did everything the same, but in place of the kit calibrant, we used the AAV9 USP reference material that I just talked about in the previous slide. And then as you can see in the table below or the graph on the upper right, 2 things are very clear right away. The absolute values are much different, even the averages. So that suggests that the accuracy of these kits the kit calibrant could be slightly flawed. And then possibly even more importantly is the precision, that once you use the USP reference material, it greatly increases the precision as compared to the different kit calibrants that come with the different kits. And this can be seen with almost a straight line or the low CV for when the test using the USP AAV9, whereas compared to the high CV for the kit calibrants. I should say this is just with AAV9. We start to see similar results, maybe a little less dramatic for some of the other serotypes, but that testing is still still being done, and we can definitely plan on sharing that in an application note when it's all complete. Next standard I'd like to talk about is the genome titer reference material. The USP developed a quantitative AAV titration PCR control for AAV genome titer. This is available at the store now, where I don't know if I mentioned it, but on the previous capsid titer products, those are not yet available, but should be available in July. So this is intended to be a standard for genome titer or Q and or dPCR methods. In this method, this product, we do 2 things. One, we have a physical product. And what's unique about this is we have 4 conserved regions on here that are very common in almost every AAV transgene. These being the SV40, ITR2, CMV, and CVA. We have in-house dPCR methods shared in the product information sheet and app notes. So we're not just providing values via dPCR, we're also sharing the method, including the primers and probes that we're using to get these values so that customers can repeat that. It's also important to note that dPCR was used for all these quantitations because dPCR is a direct measurement, not relying on a calibrant. What we hope this product can be used for, we hope it can normalize a lot of genome titer testing, and either for dPCR be used as a positive control to ensure that the values you're getting are indeed accurate, but also as a calibrant for qPCR. There's studies out there that have shown that a lot of qPCR calibrants, especially those that use plasmids, are quite inaccurate. And by having an actual AAV calibrant that was measured via dPCR in a multi-lab study, that could really help increase the accuracy of qPCR methods. Next, we're going to go to the impurities. As we talked about that, there's a lot of safety concerns with some of the impurities in the AAV, and it's very key to show that you're getting all anything out of the final AAV product that could be of safety concern. And the two products we have in this category are residual plasmid DNA and residual host cell DNA. There's a number of papers out there that show that residual DNA can pose a major risk in AAV. I highlighted 4 here, but there's a lot more out there if you ever go and look in the literature. And I'm going to highlight 3 key risks associated with that residual DNA. One, it could be incorporation into the capsids. This would cause you to have a incorrect transgene or a transgene that has a wrong sequence. It could also transfer antibiotic resistant genes to patients because a lot of starting plasmids have that antibiotic resistant genes, that there's a way that could actually get incorporated into patients. And lastly, it has an unintended immune response, which has been shown to have an adverse effect on patients. The first plasmid or the first product we developed is reference material for residual plasmid analysis. So as was highlighted in the previous slide, it's critical to ensure that you have no residual plasmid in your final AAV product. Typically, this is tested via a PCR method, and typically analytical labs go after common antibiotic region or antibiotic resistance regions, I should say. So we developed a product that has both a neo-R can-R as well as an amp-R antibiotic resistant markers on it. And then, so then anyone who targets any of those in their residual plasmid method can then use this. This is worth noting if they are targeting other regions, this will not unfortunately work. But we are planning on increasing our offerings here and generate other plasmids with other common conserved regions. So as I said, this product contains 2 antibiotic resistant genes. Its value, similar to the genome titer one, was assigned by dPCR, and we envision it being used as the same way, either as a positive control for dPCR or as a calibrant for qPCR. And in this product we have, which is also available, we include the sequences for all the primer and probes as well as the dPCR method we use. And the purpose for this is just for transparency so that anyone who is trying to repeat the results we got can easily repeat it and they can use the same exact method. Here's the data from a multi-lab study that we did that were used to assign the value for this. As you can see, 5 different labs were done, and we actually had 2 methods, one that targeted the AMP region and the other that targeted the neo-R can-R region. And I'll let you guys go through these numbers. I don't have to go through them yourself, but one thing that's really cool and really highlights the effectiveness of the methods we use is that the averages are exactly the same. And even for all 10 values from 5 different labs, the total CV is under 10%, which really highlights that we really trust these numbers and they are indeed accurate. So if anyone's interested in this product or would like to learn more about it, the product is in the store. I think I listed the catalog number, but here's a QR code. We also have released an application note on it, which goes into more detail about some of the key highlights I just talked about, and the QR code for that is shown here. Other impurity product is this one's for residual host cell DNA. As compared to the other one was for plasmid DNA, this one's for host cell DNA. We have 2 products that would be relevant to AAV users because one for a quantitative standard for HEC293 genomic DNA, and the other one a quantitative standard for SF9 genomic DNA. Similar to the other ones, we provide a lot of information in an application note, primer, probes, values, all quantified in a multi-lab study. And anyone's interested to learn more about this product, which was co-developed with ATCC, as you can, as I think I mentioned in the slide at the beginning about the timeline, but as you can also see in this application note, both these products were co-developed with ATCC. We have a lot more information available about these on. We have a whole website dedicated to this. The link's right there if you want to take a second to write that down and screenshot it. But some things in there, there's a webinar you could listen to that's specifically devoted to this. Also application notes. There's one specifically for HEC293 genomic DNA and there's another one for SF9. So anyone looking to learn more about this or interested in this product, everything you would ever need would be in this link shown right there. The last category is the raw material standards, and there's two more that fall into this bucket. One for endonuclease qualification, the other one for plasmid qualification. So endonuclease is a critical starting material. It's an enzyme, and where the idea for this standard, which is both a documentary reference standard as well as a physical reference standard, came from stakeholders saying that they don't get um, valuable information from the activity on a lot of endonuclease because different labs or different manufacturers are using slightly different methods. What we're trying to do here is generate a harmonized endonuclease method. Anyone doing this method now won't be surprised by it. It's going to be the typical method that people are doing now, including shear genomic DNA, precipitation, and ultimately a UV method. However, this method, very small differences can cause great changes. And by generating a USP compendial chapter for this, we can harmonize the way everyone does this, and then all the reportable values will make it much easier for people to compare endonuclease values. So in addition to this general or this compendial less than 1,000 chapter, we'll also have a physical reference standard that will be used as a system suitability for assessing the quality of an activity of endonuclease. Last product I'll talk about today is plasmid. This will actually, we're going to start with 6 different plasmids at different sizes. So talking to stakeholders, they felt there wasn't good reference material out there for assessing the quality of their plasmid starting material. And over the last, I'd say 5 to 10 years, more and more information has really been come to light about showing a direct relationship between the quality of that plasmid and the final product. What we decided when making this product, we needed something of the appropriate size. So that's why we decided to generate 6 products ranging in size from 4 to 14 kilobase pairs. It needed to have the right conformation. So we have these at different amounts of supercoiled so that people can assess their analytical methods. And lastly, proper topology assessment. There's a number of different methods, namely CELIF, agarose gel, or HPLC with UV detection. So we're using all 3 of these methods to look at the topology, and we'll report the values for all 3 of those on the certificate. These are not yet available, probably won't be available till the fall of 2025, but here's a quick look at what we're offering. And as I said, they vary in size. There's 6 different products, and the product information sheet will include information such as concentration, topology measurement using each method, the sequence, and identity testing to ensure that we have the right plasmid. All right, so just to wrap it up, I want to talk about a few more additional resources relating to AAV that USP has to offer. 2 general chapters, which are best practice. The top one being best practices for AAV. This focuses on all aspects of AAV from production all the way down to like the analytical testing, formulation, stability testing, whatnot. It's everything you need to know about our AAV guide. And this is currently available in our PF51-3. So we highly encourage anyone who is interested to go on our USP PF. This is free to anyone. You don't have to have a subscription to USP and to go through and make public comments on this. It's free for everyone up until July 30th. And I can assure you that every single comment, it's a requirement that every single comment is addressed. So after these 90 days where it's available on PF, the expert panel who drafts this chapter will come together again and address every single comment to ensure that the voices of the people are heard. Lastly is a 1040, which is kind of the similar, but a best practice for plasmid DNA as a starting material. I talked a little bit earlier about the importance of plasmid DNA, and this is kind of an all-inclusive guide for plasmid DNA as a starting material. Talks about manufacturing it, some of the analytical tests for it. This has already been in PF. We received over 400 comments, and this is expected to be published in USP NF and become official end of summer or early fall. Lastly, we have a USP gene therapy guide I talked about. It provides appropriate analytical tests and USP standards in one location. Anyone interested in this, I strongly encourage you just to go through it. I could spend half hour going through it all, but it's a very easy to use website. Website's here, QR codes there. Check it out, play around. So my final slides, there's a lot of different ways to get involved with USP, from round tables, donated method, being part of collaborative studies. We highly encourage anyone to go out. We have some links here. I'll have my email address in the next slide. Reach out to me. So anyone who is interested at all in getting involved with some of the stuff USP is doing, we are always looking for different collaborators. So strongly encourage you to reach out. Don't hesitate at all. Your interest will be well received. So thank you everyone for your time. As I said, here's my email, my first dot last name at usanthony.blaszczyk@usp.org. So thank you for your time. I really appreciate it. Happy to answer questions later, but this time I'd like to pass it off to the next presenter.
Okay, so hello everyone. My name is Paul Getty. I'm a senior technical specialist, and I'm joined by my colleague Lauren Tomlinson, who is a scientist. We both work for Pharmaron, based at the Liverpool site in the UK. And we're going to be talking about the analytical characterization of AAV content throughout the manufacturing process, utilizing mass photometry and orthogonal techniques. So to start, I'd like to give a little bit of an overview of the company. So Pharmaron is a fully integrated global pharmaceutical service provider with over 20 years of operation and manufacturing experience. We have more than 21,000 employees worldwide, with sites in China, US, and the UK, and over 2,000 customers. We serve the top 20 global pharmaceutical companies and provide end-to-end R&D and commercialization services. Our mission is to support partner success in discovery, development, and commercialization of innovative medicines. We offer a complete gene therapy development solution covering bioanalytical and preclinical services in the US, and offering plasmid, cell line, vectors, and state-of-the-art analytical characterization in the UK. So the Liverpool site is a world-class centre for gene therapies. We offer CMC development and manufacture of AAV at up to 500 litre scale using our platform approaches, including our in-house gene of interest backbone design, suspension-based cell line, and chromatography-based purification processes. We also manufacture both research and GMP-grade plasmid and product here. So a bit about AAV. So the adeno-associated virus is a small non-enveloped parvovirus. In recombinant AAV, the rep and cap genes present in the wild-type virus and responsible for viral replication are replaced with a gene of interest designed to have a therapeutic effect in the patient. Capsids are made up of 60 proteins. The subunit names are VP1, 2, and 3, and it can package up to 4.7 kilobases of genome. This is ideally the gene of interest that's packaged, but in practice, the situation is more complex and requires a rational application of supporting analytics to elucidate. So some of the challenges from AAV and where it differs from conventional biotherapeutics is in its complexity. So AAVs are large. They measure in the megadalton range, and the capsid itself has multiple levels of heterogeneity linked to both the actual stoichiometric composition of VP1, 2, and 3 in that 60 subunit capsid, and the fact that each protein can have a different post-translational modification profile. These PTMs may contribute to product efficacy, stability, and safety. In addition to this, the packaging of the DNA is found to be heterogeneous, with some capsids not containing any payload and others containing more than expected. Batches are much smaller than conventional biotherapeutics, and concentrations tend to be much lower, meaning that method development must proceed with much less material. To address these challenges, we offer an extensive panel of analytics covering the various characteristics shown on this slide, taking an orthogonal approach from an early stage. We offered these methods at the Liverpool site with the exception of cryo-EM, which we work with an outsourcing partner to cover. The rest of this presentation will focus mainly on the measurement of capsid packaging heterogeneity, so that is the empty-full ratio, and I will start by introducing our most recent addition to this category, mass photometry. So mass photometry is based on interference reflection microscopy and interferometric scattering microscopy. The sample is illuminated with a light beam. Some light is reflected and some is scattered. The interference between the reflected and the scattered light produces a signal known as a mass photometry contrast. This contrast is proportional to the mass of the analytes. With a mass calibration, we can then determine the masses of all species present in our sample of interest. One of the first things that we did when we got the mass photometer on site was to look at mixing in different proportions empty and full standards of AAV. So the empties were comprised of mainly empty capsids, the fulls were fully packaged capsids. And we plotted out the relative amount of empty species for the expected versus the observed. So on this plot here, you can see how much empty we would expect to be based on how much empty sample was in the mixture, and how much we actually observed is represented on the y-axis and measured by mass photometry. And as you can see, this results in very good linearity. And we've repeated this study across a number of different serotypes, and each time seen really good linearity of response. Okay, so in this slide, I've provided an overview of our manufacturing process. We have a platform approach built around a suspension-based HEC-293 cell line. Scale-up proceeds via shape flasks and seed bioreactors and ends in a 250 or a 500 litre bioreactor. This part of the process takes between 2 to 3 weeks. Then triple transfection of the gene of interest, the rep cap, and the helper plasmid is performed to initiate the production of the AAV, which takes about 2 to 3 days. Harvest proceeds via either a lysed or a non-lysed approach, depending on the serotype in question. We are a multi-serotype and multi-product facility. The harvest material is then put through clarification, affinity purification, and chromatography-based polishing for the removal of product-related impurities. The material is then concentrated and exchanged into final formulation to produce drug substance. In order to support this, we deploy a wide range of analytical techniques across this process. In the next 3 slides, I'll take a look at how the application demands the selection of an appropriate analytical approach. Okay, in this slide, I've included a table that lists a number of parameters to consider when selecting an analytical approach. So that's speed, sample number, partial species resolution, volume requirements, and validation requirements. I've included some orthogonal methods for assessment of empty and full. So there's mass photometry, AUC, and SECMALs here. So the color scheme indicates whether the approach satisfies each of the parameter requirements. So where we have a blue, this means that the requirements are satisfied, and where we have orange, this means that the requirements have not been satisfied for the particular method. In this example, I'm looking at the measurement of harvest material to support an upstream development study running at the amber scale. So we would typically clarify the harvest material and put it through some small scale affinity purification ahead of empty full determination. The separation of partial is important, as partials tend to behave more like full than empty during purification. So if the partial species are considered as an impurity, this means it's less likely to be cleared with the empty capsids, and it's more likely to be retained and at last be present in that final purified product. So in a sense, we know that there is some sort of impurity present in that product if we can resolve the partials from the fulls. So in this case, the study we're looking at is trying to reduce the initial levels of partially filled capsids, and therefore that information would be critical to the success of the study. And based on the performance of each of the methods, we can see that where AUC struggles is in terms of that turnaround time. So if we have a high number of samples, which we might if we're running at this amber scale, then it might take a very long time to get results for them all to be returned based on the throughput of the method and the time to result. The volume requirements for AUC are also very high. So at that sort of a scale, we need to commit quite a lot of our material to just this one analytical test when we might have to run many other tests alongside. SecMal struggles here in terms of being able to resolve partials from fulls in that it simply can't. Okay. So this application is based around performing in-process control testing during the AEX chromatography step designed to enrich the full material. Samples are taken from chromatographic fractions in order to confirm the pooling strategy to obtain optimal yield and purity. Speed is of the essence here. So MASS-P allows for a very rapid turnaround of results with very little material being committed. And in this example, we're looking at IPC testing for an early phase product, so formal validation is not required. This would come into play if we were looking at this type of testing for a late phase product. AUC doesn't support the sample turnaround time required for this type of testing, taking at least a day for time to result. SecMal again doesn't perform in terms of the resolution of those partial species. In this final example, we're looking at release testing of drug substance. The key determinant in our choice here is driven by the requirement for formal method validation. Our current validated offering at the Liverpool site for empty full testing is AUC. We don't currently have validation in place for either mass photometry or SecMal. That's not to say that the methods can't be validated. SecMal, though, still doesn't meet the parameter requirements in terms of being able to resolve out those partial species. So I hope these examples have helped illustrate how we assess and deploy analytical methods to meet the requirements of particular applications. In the next part of the presentation, my colleague will be talking through a couple of case studies that we have performed.
So I'm Lauren, and I'm going to talk you through a couple of case studies of work carried out at Pharmaron. So our first case study is AAV serotype and transgene analysis of commercial bought-in material. So as the manufacture of AAV results in the production of a range of capsid species, shown here in the top left figure, species can include empty, which has no payload, truncated or contaminant genomes known as partials, full, which contains the intact gene of interest, and overfull, which is larger than expected genomes. So capsid species that are not fully packaged are considered as product-related impurities, and these can impact on clinical efficacy, safety, and the immune response. So analytical techniques are required to quantify relative levels of empty, partial, full, and overfull species to inform process development and then to quantify the relative purity of clinical or commercial material. And we can use MASS-P to determine what size a particle is. However, we do need to understand what is in the particle. So, for example, an impurity could be the same size as a partial species. So we have ddPCR to inform us that the gene of interest is in the product by the titer determination, but we don't know which one it is. So it could be from the partial, the full, or the overfull species. So we have other methods, including CE and NGS, which can look at the genomes that are in the sample and give a more in-depth analysis as to what is actually packaged in the product. So for this case study, we've used two serotypes of two different gene inserts, and they were analysed by mass photometry, AUC, NGS, and CE to determine the AAV content profiles, ranging from empty to overfull species. So here we have the complementary data from MASS-P and AUC on the AAV5 serotype with two different gene inserts. We have GFP on the left and LAC-Z on the right. So for the MASS-P, the x-axis is in mass in kilodaltons, and for the AUC, the x-axis is sedimentation coefficient, and this is proportional to mass. So for the GFP data, we see three species, and based on the masses of each species, the first region matches up to empty. Second is a capsid plus gene of interest, which we term full. And then finally, the third region is classed as overfull, which is consistent with an empty capsid with two genes of interest inside. For the LAC-Z insert, we also have 3 species. We have the first region, which corresponds to an empty mass. The third region is consistent with a capsid plus a gene of interest, which this is larger in mass compared to the GFP due to the larger size of the LAC-Z insert. And then the region in the middle of these two is termed as partial species, as the mass of this region is too big for empty and too small for the full species. So the complementary data provided by the gold standard method of AUC shows good agreement to mass P for the percentage of different capsid species. And for this AUC testing, we use just one replicate, as this method needs more sample volume, whereas the mass P uses little sample volume, so we can do triplicate analysis and get good precision data. So if we're moving on to the additional serotype, we have here the data for the AAV2 serotype with 2 gene inserts alongside the AAV2 data. And between the 2 serotypes that we've chosen to analyse, we have every possible region covered from your empty to the overfull species. And for both the serotypes of the GFP insert, we have empty, full and overfull. And for AAV5 LAC-Z, we have empty, partial and full. And for AAV2 LAC-Z, we have partial and full species. So by utilising mass P, we can successfully resolve the different species from these different AAV serotypes and transgenes. So with mass P, we can achieve resolution of 3 species, but we need to identify what is in each of these species. So the smallest species is consistent with an empty mass. However, the other species are indicative of genome inside, and so we need alternative methods to determine what exactly these are. So we can use capillary electrophoresis coupled with laser-induced fluorescence, which allows for rapid and sensitive characterization of the AAV genome size and integrity. So the sample preparation involves genome release and sample denaturation prior to labeling with a fluorescent dye. And this method uses low sample requirements at only 50 microliters of sample. So we can see how the sample migrates against the capillary and compare this against the DNA ladder to determine the genome length. So the theoretical size for the GFP insert is 2.4 kilobase pairs, and we see a peak within the region of 2 to 3 kb, which suggests that this is GFP. We can also use long-read NGS, which analyzes the AAV payload directly and matches against the nucleotide sequence of the gene. So it can detect changes in the DNA sequence and also detect structural differences, such as truncations and rearrangements. And the results can provide a qualitative abundance of full and partial, but it can't quantify the empty. So for the NGS data, which is not fully optimized for this product, but we're just giving some example data, we predominantly see one peak that corresponds to the size of GFP, and additional species around double the size of GFP is also observed, which could suggest that it is a concatenameric species of GFP, where the 2 GFPs are joined together by a link. And we also have some lower species present, which could be some truncated GFP that has broken apart during the testing process. So for this case study, we have shown how useful orthogonal techniques of NGS and CE can give us additional insights into the identity of the overfull species that was identified by the mass P. And moving on to case study 2. So now we'll be looking at how mechanistic modelling can be used during the AAV purification process. So we know that there will be impurities in the process, and we need to remove these to increase the purity and the yield of the product. So we use an AEX polishing step for separation. However, this step can be quite difficult to separate the partial from the full, as they are quite similar in terms of their mass and their charge. So we need lots of experimental data to feed into the model and characterize the system by running samples on the column to gain data from lots of different fractions using a good precision method to resolve the empty, partial and the full species. So the AAV regions of empty, partial and full are then overlaid across the AEX chromatogram to determine the most optimal conditions, and the model can reduce the amount of time needed to optimize the process development of the product. So in order for the modeling to be successful, we need an analytical method that can analyze data from many fractions with high accuracy and precision, and that can resolve empty, partial and full species from each other. So this is where we can utilize the MASP for the analysis of the fractions to determine the empty, partial and full content. So here we have 19 AEX fractions showing the proportion of empty in the dark shade blue, partial in mid blue, and full is in the lighter shade of blue. So this is not an optimized process for this particular product, but we're just using material that we know contains empty, partial and full to help generate the model. So by using MASP to analyze these fractions, we can get data quickly with good accuracy and precision using very little sample volumes. And here is the AEX chromatogram showing absorbance at 260, which is DNA, and 280, which is protein. So the first peak has more absorbance at 280, as there are more empty capsids. There is also absorbance at 260, as the absorbance of proteins will also absorb in this region. So the second peak, which is covering more of the fractions, switches to more absorbance at 260 due to these fractions containing more full species, and the 260 absorbance correlates to DNA in these species. So as we see the number of fractions increase, we can see a decrease in the number of empty and partial species shown in the darker blue shades, which suggests that we are removing more of these impurities. So the advantage of using MASP to analyze these fractions is that they can be acquired in a single experiment. So if we were using AUC for this, we would require multiple AUC runs and more sample material, whereas all of this analysis can be completed within 1 day using the MASP, which really highlights the importance of selecting the correct method for the required type of analysis. And just to conclude, so here at Pharmaron, we can utilize different analytical techniques based on the key requirements of the study, so such as the parameters of the concentration of the material, the sample volume, analysis time, and regulatory compliance considerations. So we have shown that MASP can be used for the optimization of AAV capsid purification by analyzing samples quickly and accurately at key stages in the downstream process. And with very little sample volume requirements, it can support many studies, including the mechanistic modeling. And in addition to MASP, we can also use orthogonal techniques, including AUC, NGS, and CE, which can provide complementary information regarding the AAV serotype and transgene insert, which then gives us a deeper understanding of the product. And finally, we would just like to thank all of these following team members at Pharmaron who have helped support the analysis for our case studies, and thank you to the listeners for taking the time to join us today.
Thank you all for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Our first question asks, what are the biggest barriers to implementing orthogonal methods in your current QC workflow? Anybody want to take this one? Thank you, Paul. You're not unmuted, or you're muted, sorry.
Oh, sorry. I'm happy to handle this question. So with this, we have considerations whenever we're moving methods into QC based around, oh, sorry, the question's moved. So essentially, in order to deploy this method into QC, we would need to go through a process of evaluation and implementation of that compliance software that I know Refine does currently offer. As well as this, we would consider whether or not we wanted to move to the automated platform in terms of the sample preparation and handling, just to make it a bit more simpler and more compliant. Similarly, we would have to assess the qualification of both software and instrument, and we would do that in concert with our validation and QA colleagues. So there's quite, I guess, a few barriers, but nothing specific to this technology really. And based on what we've seen so far, we do believe it's something that's worth pursuing.
Wonderful, thank you. Our next question here asks, how does the inclusion of MP in the USP General Chapter influence its acceptance or adoption within industry or regulatory agencies?
So in terms of acceptance within industry, I think we're showing its value, including in our certificate for empty and full standards, including in our general chapter or analytical guides, as being more visibility. In terms of regulatory, it's not going to help much there. The biggest impact with regulatory would be a donation of a validated method and get it into one of our compendial chapters, which is less than 1,000. I don't think I discussed those too much, but those are recognized by regulatory agencies, and at that point, people can kind of plug and play, only do a verification rather than a full validation. But at this point, we don't have actually any empty full methods, including mass photometry, in any of our less than 1,000 chapters. But hopefully, it's something we can move towards in the future.
Excellent, thank you. Okay, our next question here asks, are there specific stages of the manufacturing process where MP delivers the most actionable insights?
Yeah, we get very good informative results during the IPC testing. So where we need to analyze fractions from AEX testing quickly to inform the next stage of the process for enriching the full material. So MassP gives us great quick turnaround, unlike something like the AUC where we'd be waiting a day then for the results. So IPC testing, and just in general on the optimization of the purification process, especially around the AEX step, where we're focusing on removing the product-related impurities. We get great informative results from that.
Yeah, and if possible, I'd like to add something to that, because I think we touched on the idea of this increasing scrutiny around these partial or intermediate species. So we find that MassP gives us very useful data in that respect. Part of the challenge with these partially filled intermediate species is that they behave very much like full capsids in terms of the polishing step, so that anion exchange step. It can be very challenging to deal with them at that point, and as they are generally considered as impurities, then we would be ideally clearing as much as possible. One thing that mass photometry allows us to do is to go very far back into the process towards the end of the upstream part, and even look at something like studies based in amber vessels, looking at different conditions, different plasmid constructs, in order to try and screen out those that might have some sort of prevalence towards generating that partial material in the first place, to remove that challenge in the downstream just by reducing it at the point of manufacturing. Because of the low volume requirements and the ability to run quite a lot of samples, we can plug this mass photometry into something like an amber study quite handily and get some very informative information.
Wonderful, thank you both. Okay, our next question here asks, how does the full capsid standard work when using different plasmid sizes? For example, if you have a smaller transgene size, the full capsid will be smaller than the one with the larger transgene.
Yeah, it's a good question. The short answer is the standard is not going to be representative of actually anyone's actual AAV, but it's meant to be used as either a system suitability or positive control. So similar to how someone might use like a standard and doing a UV measurement to ensure that your absorbance is in the right area, you could use the USP empty full standard to ensure that whatever method you're using is that day, at that time of measurement, getting either the right molecular weight, right size of that particle, and even most importantly, the percent empty full. So it's more of an analytical tool to ensure that your method and analytical instrument is working as intended. So that's why we see it more being used as a positive control. Um, just the idea of having representative standards for everyone is just never going to be possible, because every AAV is always going to be different.
Wonderful, thank you. Okay, we're going to try to squeeze in a couple more questions here. This next question asks why secmols is considered accurate regarding measurement of capsid titer, and is there any special reason, meaning which method is more accurate regarding capsid titer and why?
Yeah, so another good question. Generally speaking, ELISA is the most used. I was just kind of comparing mols to ELISA, and why mols is preferred is because it's a direct measurement. ELISA is always going to be an indirect measurement, relying on that calibrant, and then back calculating your titer based on that calibrant. But possibly even more importantly, when looking at specifically AAV ELISAs, most of those calibrants are themselves measured by an indirect measurement. So you're almost getting like a third tier measurement when you're doing an ELISA. So if those ELISAs are doing a direct measurement of the calibrant, it'd probably be much better. But most of the time they're doing some type of either, for example, genome titer and back calculant based on percent full, which is just, there's too many variables coming in. SEC mols, we're measuring the extinction coefficient ourselves of both the nucleic acid and capsid titer, and directly measuring it on that instrument. So it's the first degree of measurement by a method that's been proven to measure pretty accurately. So we don't have, ultimately you're gonna have to go through all the guidelines to assess accuracy and everything, but we just feel most comfortable with that method because it's, as I'm repeating myself now, it's a direct measurement rather than indirect measurement relying on calibrants.
Excellent, thank you. And we have time for just one more question here. So our last question of the day, any challenges when using clarified bulk harvest using MP?
I think the the biggest challenge there is just the continued presence of host cell related impurities that are going to show up down at the lower end of the mass range, and you do tend to see a bit more signal there with this type of material. You don't typically see anything that overlaps the mass range, even encroaching up as far as the empty or the full, in my experience. But one thing we do look at is aiming for an affinity purified sample to measure. So we can do that by having a scaled down affinity purification step. So either like a plate-based format or a bead-based format, or a mini column on a biomech or something, functionalized with AVX on the relevant affinity ligand, and essentially pushing those samples through that scaled down affinity step to help clean them up a bit further if we're struggling to get the data.
Excellent, thank you. I want to thank you all for your informative presentations. We would also like to thank LabRoots and our sponsor Refine for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.